So we're going to look at exceptions to Mendel's rules today, but first review what Mendel actually found from his studies. So everything that Mendel studied and discovered is true. It's just only true to the things that he studied. And there's lots of things he wasn't able to look at. So first, let's look at what he did. Remember, he studied pea plants. He said that these things that were inherited or passed down were called factors. We now call them genes. They had very limited information at that time in the 1860s about chromosomes and DNA. He said that every gene had two forms. We now know that that is not true for everything, but everything he studied did have two forms. And those factors, if you got one of each form, those factors, according to the principle of dominance, basically said that the dominant allele would be seen, the recessive allele would not be seen. We also see in the Punnett square the law of segregation, that the alleles are separated and they're independently separated. They're not connected. And we see that as we do our Punnett squares, and that's what we're showing here, is that separation and that independence as far as when they recombine and depending on which one gets fertilized. This is called a monohybrid Punnett square. We only looked at one trait at a time up until now. Today you got to look at dihybrids. And in dihybrids, you're looking at how two traits are inherited and the likelihood that those two alleles will show up together. What we've seen so far is called complete dominance. So the capital letter stands for the dominant trait. That's what you see. The recessive only shows up if the capital letter is not present. So that's the one you don't see. Now, exceptions to Mendel's rules, lots of things that were discovered after his work and after he passed away. And we're going to look at some of these today: incomplete dominance, codominance, multiple alleles, polygenic traits, X-linked traits, and pedigrees, which is how we can study genetics in humans. So incomplete is exactly what it sounds like. It doesn't completely get expressed. So it's okay if you have the homozygous, that will be black. If you have the homozygous W, that'll be white. It's when you have the heterozygous, you're going to see this incomplete. So the alleles are just going to be partially showing. So in this case, it'll look gray. So another way to look at it is a cross between two organisms with different phenotypes, that there's a blending of the parental traits. If you look at snapdragons, these flowers do the same thing. And if you have a homozygous red, it will be red. Homozygous W, it will be white. But if you have one of each, one R and one W, it's going to be pink. And we'll look more at that when you're doing some example problems. Codominance is another exception to Mendel's rules. Again, if you have the homozygous, it'll be black. In the homozygous W, it'll be white. But if you have both a B and a W allele, it will actually be speckled. And in chickens, we call this erminate. It'll actually be speckled black and white. From a distance, it may still look gray, but it's totally different because both things are expressed equally. So here's the example of the chickens. Notice again, we're going to have to write them differently because capital and lowercase letters will no longer make sense. In cows, you have white cows if you have homozygous W. You have red cows if you have homozygous R. And if you have one of each, they're called roan. You can see those are the ones that look kind of this reddish or muddy brick red or brownish and white spots. Blood typing is another example of codominance, but it's also another exception to Mendel's rules. Remember that Mendel said there were only two options for each gene. We're going to see that there's three or more in many different examples. So if you're looking at your blood type, you actually have three different letters. You have IA, you have IB, or you have little I. Now you can only still have two of those, but which two you have all these different choices. So that's multiple alleles. Polygenic traits are where you have a spectrum. So you have multiple genes that end up influencing a trait, and those multiple genes will cause more of a spectrum. So that's a hair color, eye